Chapter 21 is on congenital and genetic disorders, and I split it into 2 PowerPoints just to make the lectures a little bit shorter. And I have one labeled congenital disorders and one genetic disorders, even though there is crossover between the two. And we'll talk about what is the difference between a congenital disorder, developmental disorder, and a genetic disorder. So genetic information is stored in chromosomes in our body. And so it's the DNA bundled up into the little chromosomes. We have 23 pairs in humans. 22 pairs of them are what we call autosomes, and 1 pair of sex chromosomes. So a female has 2 X's and a male has an X and a Y. So a karyotype is a visual representation of chromosomes arranged in order of size. So whoever first came up with this, when they arrange them, they put the biggest ones first. So number 1 are the longest, and number 22 were the shortest, and then X and Y are at the end. So karyotypes are used in the diagnosis of chromosomal disorders. So during meiosis in humans, each of the gametes receives half of the chromosomes, half of each pair. There is a little bit of crossing over of genetic information, so you might not have exactly the same 1 chromosome that your mother or your father had. But then when the ovum is fertilized, you get a new set of 23 pairs. So it's pretty neat. Like, I always like to say that my sisters and I are 3 iterations of the same genetic experiment, and we all came out very similar but a little bit different. So like, mix the same chromosomes together and see what you get. It's kind of interesting. This is an example of karyotypes from the book. The 2 on the top are normal, a female on the left and a male on the right, and the 2 on the bottom have some disorders that can be identified in the karyotype. So the one on the bottom left is a trisomy 21, which is Down syndrome, and it's a male. You can see there are 3 of the 21 chromosomes and an X and a Y. You can identify it as a male. On the bottom right, it's someone who has a translocation between 14 and 15, and that causes problems as well, and we'll talk about why a little bit later. So a karyotype can be used to diagnose certain disorders, but not all genetic disorders show up in a karyotype. A lot of times the changes occur in our chromosomes because of an error in either meiosis or mitosis, but that's relatively rare that happens. Sometimes you get a mutation that is, or an alteration in genetic material that's spontaneous, or it's a result of exposure to harmful substances such as radiation or chemicals or drugs. So there's a lot of, in the last 20, 30 years, a lot of research has gone into mapping chromosomes, mapping genes, and figuring out what parts of the genes do what, with an eye towards treatment of genetic disorders later on. So we talk about genotype and phenotype, and genotype is the actual genetic information carried by the individual. So all of the cells in your body except the gametes have the same genotype. So whether it's a liver cell or a neuron or a skin cell in your pinky toe or any other cell in your body except your gametes, we have all the same genotype, but not all the genes are expressed in all the cells. So it's a little bit of a mystery as to how that happens in development, that some cells develop one way and some cells develop another way with the same genetic material. But I kind of like that there's a little mystery still out there. Maybe someday we'll know how it works, but it's mysterious right now. So that's the genotype. That's the information, the genetic makeup of all your cells. The phenotype is the expression of the gene. So it's the actual physical manifestation of an inherited trait, or in the case of genetic diseases, the manifestation of the disease. So it's how the genes are expressed and the individual's characteristics. So I like to say, just as a way of remembering it, the genotype is what you're dealt, and the phenotype is how you deal with it. So our genes do control all of our physical characteristics, such as eye color. That's always an example that's used, but also all of our metabolic processes, how we heal, how we form scars, how our connective tissue works. So that can affect a lot of processes in our body. The effects of the genes vary with gene penetration or frequency of expression of the gene. So they'll talk about genes having low penetrance or high penetrance. High penetrance means that if you have the gene, it's usually expressed. Low penetrance means it may or may not be expressed. So there are a lot of traits like eye color and blood type and a lot of different things that are influenced by more than one gene. So the phenotype is going to vary a lot depending on how the genes are expressed. So congenital disorders are disorders that are present at birth. So this includes genetic or inherited disorders as well as developmental disorders. The difference being that genetic disorders or inherited disorders can be passed on to the next generation, and developmental disorders cannot. So inherited disorders can be caused by the expression of a single gene. They can be caused by a chromosomal defect. They can be caused by multiple genetic expression, so polygenic expression, lots of genes contributing to the effect. Single gene disorders, the trait is controlled by one set of alleles, and it's transmitted to subsequent generations. So I have the relax, nothing is under control, meaning we cannot control what genes we inherit from our parents or what genes we pass on to our kids. So, with the advances in research, perhaps there will be some treatments for genetic disorders on the horizon, but at this point, you're kind of stuck with what you get, unfortunately. So chromosomal anomalies result from an error usually during meiosis when the DNA fragments are displaced or lost, and it alters the genetic information. So Down syndrome is an example where you end up with 3 chromosome 21s. You can also get translocations where you get abnormal placement of a part of a chromosome, an altered structure like a deletion where part of the chromosome just gets left out. And so that is an error in chromosomal duplication. So these type of defects are more common when the mother is older than age 35. And there's also new research to indicate that there's a higher risk in children of older fathers as well. So as we age, which we'll talk about in this module as well, things are not working at their optimum. So there can be more errors in meiosis. So chromosomal anomalies are found in approximately 7 in 1,000 births. So that's fairly frequent. But other congenital or developmental disorders result from premature birth, difficult labor and delivery, such as CP. A lot of times that's the baby is deprived of oxygen during a difficult labor and delivery, or exposure to a damaging agent during fetal development. So the defect might be limited to one organ, such as the heart, or it may affect the function of many organs. Congenital disorders like this do not have a genetic component, but because they manifest at birth, they're termed congenital or developmental. So developmental defects might be spontaneous errors in the DNA, or they might result from exposure to environmental factors in utero. So in the pharmacy chapter, we talked about teratogenic agents, teratogenic drugs, but there are other things besides drugs: alcohol, hormones, cigarettes, German measles or rubella, exposure to lead and mercury. All these things can cross the placenta and affect the developing fetus. So because the mitosis and differentiation are happening rapidly in a developing fetus, these changes can have a profound effect in the first months of embryonic development. So the teratogenic agents cause damage during that developmental period. There are multifactorial disorders, and a large number of disorders are of this type. They often have patterns of familial inheritance, but there's an environmental component as well. So multifactorial disorders are estimated to affect approximately 10% of the population. So they might be polygenic, or they may be the result of an inherited tendency towards a disorder that's expressed after exposure to certain environmental factors. So we talked about this early on in the development of pathology. You might have a predisposing genetic makeup, and then you have a precipitating environmental component. So genetic disorders really have a lot of social and psychological implications. Deciding whether or not to have kids, if there's a risk of such disorders. So there, the other thing too is you have to think about when you do have a child, if you have a history of certain things in your family, how is that going to affect their life? What accommodations are you going to have to make? So there are lots of decisions to be made with regard to genetic disorders and congenital disorders. The congenital or developmental disorders that result from premature birth or difficult labor and delivery, cerebral palsy is a big example of that. A lot of times we will be working with people with CP in therapy. Since I don't work with children, I primarily work with adults. Usually when I'm working with someone with CP, it's an adult who's had CP their whole life. And so a lot of times they've been through lots of therapy. They kind of know what's going on. So you could be working with someone on all sorts of different things: balance, range of motion. With children, a lot of times you might be doing serial casting or serial bracing, where you're working on straightening out limbs. You might be working on coordination issues. So someone with CP is someone that we see a lot in PT throughout their lives. So it's really interesting. I've worked with some really interesting people with CP with different levels of disability, from people who were very independent and able-bodied, and people who were very dependent and not able-bodied. So chromosomal disorders, I'm sort of doing things a little bit out of order from the book just because the order makes more sense to me. Chromosomal disorders are usually caused by an error in meiosis. Down syndrome is a classic one, trisomy 21. It may be caused by non-disjunction or translation during meiosis, and is usually when you have an older mother. The Turner syndrome, it's XO syndrome, where the person has an X but not a second X, or no Y either. When you just have an X and no Y, you turn out as female. And usually Turner syndrome females are of short stature and they are infertile. Klinefelter syndrome is sort of the opposite. 2 X's and 1 Y, there's an extra X chromosome present. Klinefelter can also result in infertility, and it frequently has developmental disorders as well. So there can be developmental delays. People with Klinefelter syndrome also a lot of times have connective tissue issues, and the few people that I've worked with Klinefelter in the clinic are because they had connective tissue disorders, and I was treating them post-surgically, like maybe they had a tendon repair or something going on. So you're not necessarily treating someone with Klinefelter syndrome and PT unless they have a subsequent orthopedic issue. So Down syndrome is a common chromosomal disorder. It's trisomy 21, and it affects physical and mental development. The risk of Down syndrome increases with maternal age, and there is a screening test that they do in the first trimester that includes ultrasound and maternal blood screening. They can screen for risk of Down syndrome, and they can also do amniocentesis or chorionic villi sampling to confirm the disorder prior to birth. So the whole idea of that is then you know what to expect and you can prepare for what you're going to have to do differently to take care of this child with Down syndrome versus one that doesn't have Down syndrome. So there's a video that I linked to in the module about a competitive gymnast who has Down syndrome. That's really awesome, kind of uplifting video. I love that stuff. So, look at it if you want to. But people with Down syndrome are capable of a lot of things. So, bear that in mind. The characteristics of Down syndrome include the physical characteristics, include a small head and a round face with a flat facial profile. I love this picture from the book. I think this little guy is just such a cutie. But slanted eyes with an epicanthic fold, a large tongue and a high arched palate, which can sometimes interfere with feeding and nutrition. Small hands with a single palmar crease. Normally you have two palmar creases. Take a look at your palm right now. You'll see you have two creases. With Down syndrome, there will be a single crease. Short stature. Muscles tend to be hypotonic and have loose joints. So a lot of times if you're working with someone with Down syndrome early on in PT when they're children, they require some extra support because of their hypotonia, low muscle tone. And so you might be teaching their parents what they need for support and how to position them correctly. Later on, you might be working with someone working with exercises for stability. People with Down syndrome typically have delayed developmental stages. The cognitive impairment with Down syndrome can range from mild to major. So it's going to be vary from person to person. They are going to have delayed or incomplete sexual development. Males with Down syndrome are infertile, and females have a lower rate of conception. Of course, this being a genetic disorder, it can be passed on to children. So multifactorial disorders are genetic influences combined with environmental factors. So some examples of multifactorial disorders are cleft palate, congenital hip dislocation, congenital heart disease, type 2 diabetes. So it's basically you have that genetic predisposition interacting with environmental factors which may or may not cause you to develop the disorder. So it's a mystery as to how they interact, but there is a multifactorial effect. So it's not just having the genetic disorder, there's also an environmental factor involved. So some of the neurodegenerative diseases, like multiple sclerosis and Guillain-Barré, might be considered to be multifactorial disorders because it's not purely genetic, it's not purely environmental. There is an interaction between multiple factors. So developmental disorders can be caused by maternal exposure to drugs, chemicals, or radiation during childbearing years. TORCH is an acronym for maternal infections that can result in anomalies. And there are routine prenatal screening tests to test for these type of infections. Exposure to known teratogens in the first 2 months of development impairs the development of organs in the developing fetus. So developmental disorders can be caused by difficult labor and delivery. They can be caused by exposure to teratogenic compounds. They can be caused by maternal infections. So this is the effects of teratogens during pregnancy on different areas of the developing fetus. A lot of times, the earlier on the exposure is in the pregnancy, if it's before 10 to 12 weeks, the more effect there's going to be. So usually exposure within the first 2 weeks results in a spontaneous miscarriage and death of the zygote and embryo. If the exposure happens in the first 8 to 12 weeks, you're going to have a major effect. If it happens later on, you will have a more minor effect. So remember that for neuro next winter. We'll talk about that again. So when you look at the development of the central nervous system, you can see that things pretty early on have major effects.